1 billion people worldwide live with headache disorders. Not occasional headaches from dehydration, not tension from a bad day. Diagnosable neurological conditions that return repeatedly, that can last hours or days, and that, in their most severe form, are classified by the World Health Organization as one of the most disabling conditions a human being can experience. Migraine is not a bad headache. It is a complex neurological disorder involving multiple brain regions simultaneously. A disorder with 4 distinct phases, each driven by a different biological mechanism, each affecting different systems in the brain and body. Today, BodyCode decodes the complete biology, from the first molecular warning signal that arrives up to 48 hours before the pain, to the wave of electrical suppression that sweeps across the cortex, to the inflammatory soup that activates the pain system, to the central sensitization that makes the entire nervous system hypersensitive. This is migraine. Before we go inside the migraine itself, we need to understand the brain it happens in. Because migraine does not occur in a normal brain that suddenly malfunctions. It occurs in a brain that is fundamentally different in its baseline excitability. Research published across multiple institutions has consistently found that the brains of people with migraine show altered excitability thresholds in the cortex compared to non-migrainers. The neurons fire more readily. The sensory processing networks are more reactive. Stimuli that a non-migrainer processes normally, light, sound, smell, are processed at a higher intensity in the migraine brain, even between attacks. This is not a flaw, it is a characteristic. A brain that is highly attuned to its environment. A brain with a lower threshold for detecting change. The problem is that this same sensitivity makes the system vulnerable to a specific type of neurological event. Now let us go to the first phase, the premonitory phase. Up to 48 hours before the headache begins, the hypothalamus activates. The hypothalamus is a small structure deep in the brain that regulates sleep, appetite, mood, body temperature, and the interface between the nervous system and the hormonal system. When migraine begins, it begins here, before any pain, before any aura. Hypothalamic activation in the premonitory phase produces the symptoms that most migraineurs recognize in retrospect, but rarely identify in real time. Food cravings, mood changes, yawning, neck stiffness, sensitivity to light and sound that arrives before the headache, fatigue that feels different from normal tiredness. These are not random. They are the biological output of a specific brain region activating in a way it should not be activating. The hypothalamus is firing the starting gun for an attack that will not fully. Now we reach the mechanism that defines migraine with aura, and one of the most extraordinary neurological phenomena ever documented in the human brain: cortical spreading depression. It was first described by a Brazilian neuroscientist named Aristides Leão in 1944. He was studying the electrical activity of rabbit brains when he observed something remarkable. A slow wave of intense electrical activity, neurons firing in a massive synchronized burst, followed immediately by a near-complete suppression of all electrical activity in the affected region. A wave of neuronal silence that spread slowly across the cortex at a rate of 2 to 6 millimeters per minute. This is cortical spreading depression. And research published in PLOS Biology in 2025 confirms it as the biological mechanism underlying migrainous aura. Here is what happens in molecular detail. The wave begins. Neurons in the cortex undergo a massive simultaneous depolarization. Potassium ions flood out of cells into the extracellular space. Sodium and calcium ions flood inward. The normal electrical gradient across the cell membrane collapses completely. In the wake of this depolarization wave, the affected neurons cannot fire. They are electrically silent, exhausted for minutes at a time. When this wave passes through the visual cortex, the region at the back of the brain that processes what you see, it produces the visual aura. The classic migraine aura is a crescent of flickering lights or geometric patterns called a fortification spectrum that begins at the center of the visual field and slowly expands outward over 20 to 30 minutes. The speed of that expansion matches exactly the speed of cortical spreading depression propagating across the visual cortex, 2 to 6 millimeters per minute. You are seeing the Now comes the pain. And this is where the biology becomes truly remarkable, because the brain itself has no pain receptors. You cannot feel your brain tissue directly. So how does a neurological event inside the brain produce pain so severe it incapacitates millions of people every day? The answer is the trigeminovascular system. The trigeminal nerve is the fifth cranial nerve. It is the primary sensory nerve of the face and head. Its branches extend across the forehead, the temples, the jaw, and critically, into the meninges, the thin membrane layers that wrap around the brain. The meninges are richly innervated with trigeminal pain fibers, and the blood vessels running through the meninges are surrounded by these same fibers. When cortical spreading depression occurs, the depolarized neurons release a cascade of molecules into the surrounding tissue. Research published in leading neuroscience journals has documented this molecular release in detail. Potassium ions, hydrogen ions, glutamate, serotonin, calcitonin gene-related peptide, known as CGRP, nitric oxide, arachidonic acid, prostaglandins. Together, these molecules constitute what researchers call an endogenous inflammatory soup. This inflammatory soup activates the trigeminal pain fibers in the meninges. Those fibers send pain signals back to the trigeminal nucleus in the brainstem. From there, signals travel upward to the thalamus and then to the cortex, where they are experienced as pain. The location of the pain, behind the eye, at the temple, across one side of the head, corresponds to the distribution of the trigeminal nerve branches. The throbbing quality corresponds to the pulsation of the inflamed meningeal blood vessels with each heartbeat. This is the trigeminovascular pathway, and according to research recognized with the Brain Prize,
now we address the symptoms that make migraine more than just pain. Because what separates migraine from other headache disorders is not the pain alone. It is the complete sensory transformation of the nervous system during an attack. Photophobia, phonophobia, osmophobia, the hypersensitivity to light, sound, and smell that makes a migraine attack so incapacitating. The mechanism involves central sensitization. As the attack progresses, the trigeminal pain fibers that were activated by the inflammatory soup become sensitized. Their firing threshold drops. Stimuli that would not normally activate them begin to activate them. The neurons become hyper-responsive. This sensitization spreads centrally, up the trigeminal pathway, into the thalamus. The thalamus is the brain's sensory relay station. When thalamic neurons become sensitized, every sensory signal arriving from the body is amplified. This is why touching your scalp hurts during a migraine, while the weight of your hair on your head feels painful, why a normally comfortable room feels intolerable. The thalamus is no longer accurately filtering and modulating sensory input. It is amplifying everything. The nausea and vomiting have a separate mechanism. The trigeminal pathway connects to the brainstem nucleus that also receives input from the vestibular system and the gastrointestinal tract. When trigeminovascular activation reaches this nucleus, it produces the nausea response that accompanies severe migraine attacks. And the autonomic symptoms, the pallor, the cold sweats, the changes in heart rate, reflect hypothalamic involvement continuing throughout the attack. The same hypothalamus that fired the starting gun in the premonitory phase is still active and dysregulated.
Let us talk about treatment, because the biology we just covered directly explains why different treatments work and why others do not. The first class of targeted migraine treatments were the triptans, introduced in the 1990s. They work by binding to serotonin receptors on the trigeminal nerve fibers and the meningeo blood vessels. This produces two effects simultaneously. It constricts the dilated blood vessels in the meninges, reducing the pulsating pain, and it suppresses the release of inflammatory neuropeptides from the trigeminal fibers, interrupting the inflammatory cascade. Triptans do not prevent migraine. They interrupt the attack once it has begun, and they are most effective when taken early, before central sensitization has fully established. Once the thalamus is sensitized, the window for effective triptan treatment begins to close. This is why timing matters. Taking the medication at first sign of headache rather than waiting until the pain is severe is not impatience, it is neurobiology. The most significant advance in migraine treatment in decades came from understanding CGRP, calcitonin gene-related peptide, one of the key molecules in the inflammatory soup released during cortical spreading depression. A new class of medications called CGRP monoclonal antibodies, approved and now widely used, works by blocking the CGRP receptor or the CGRP molecule itself. This prevents the inflammatory cascade at a key molecular step. These are the first preventive migraine treatments developed specifically from migraine neurobiology, not repurposed from other conditions, designed specifically for this mechanism. They have reduced monthly migraine days by 50% or...
There is one final phase that most people have never heard of, the postdrome. After the headache ends, the attack is not over. The postdrome phase, which can last up to 48 hours, produces a specific cluster of symptoms that migraineurs describe as the migraine hangover. Fatigue, cognitive slowing, mood changes, difficulty concentrating, a persistent sensitivity to sensory input. The mechanism involves the slow recovery of the cortical regions affected by spreading depression, the gradual normalization of the trigeminovascular system, and the resolution of the central sensitization that built during the attack. The brain has been through an extraordinary neurological event. The recovery takes time. Now consider what migraine biology tells us about what triggers attacks. Any factor that lowers the cortical excitability threshold or facilitates spreading depression or activates the trigeminovascular system is a potential trigger. Sleep disruption lowers the threshold. Hormonal changes, particularly the estrogen drop before menstruation, lower the threshold. This is why migraine is 3 times more common in women than men, and why many women find attacks concentrated around their menstrual cycle. Dehydration, fasting, stress and the cortisol response, sensory overload, barometric pressure changes. None of these cause migraine in a non-migraine brain. They lower the threshold in a brain that is already at the edge of that threshold. Understanding your own trigger pattern, the specific conditions that move your brain from subcritical to threshold, is not pseudoscience. It is applied neurobiology. 1 billion people live with this. Most of them have been told they have a bad headache. Very few have ever been told what is actually happening inside their brain. Now you know. If this video gave you something that changes how you understand what your brain is doing, share it with someone who has been dismissing their migraine for years. And if you want to keep decoding the biology behind the conditions that shape how we live, subscribe to BodyCode.